Really a great pleasure to welcome Michael as our very first inaugural distinguished lecturer for our new School of Biomedical Engineering at the University of Sydney. Michael's, as you can see there, from the Allen Discovery Centre at Tufts in Boston. And he's very well known for the discovery of xenobots, which are described as living robots that are made from frog skin cells that can sense their own environment. And as an engineer, I think it's amazing that this discovery was simulated first and then built. That's how we really like to do things in engineering, and a great use of biology, synthetic biology. So Michael's got a great treat for us, and I'd like to welcome him to start his presentation. Thank you, Michael.
Thank you so much. And yeah, I'm very honored to be able to share some thoughts with all of you. If anybody is interested in getting in touch with me later on, you can find all the primary papers and everything else at these websites. What I'd really like to talk about is cognition and how minds can exist in the physical world. I want to make sure, however, that it doesn't just stay at the level of philosophy. And what I'm going to do in this talk is something a little unusual. I'm going to mix and sort of jump back and forth between some of the conceptual theory that we've developed around that idea and practical applications, because in our group, these form a very tight feedback loop where we develop theory and then actually test these in the lab in biomedical contexts. So I'm going to go back and forth between some fairly philosophical subjects and some very practical regenerative medicine approaches. So I think that everyone will recognize who this is. This is Alan Turing, and we know him as one of the forefathers of artificial intelligence. He was very interested in what minds are and how we can create novel minds. Actually, we already create minds all the time. Those would be children. But this is what Dan Dennett calls competence without comprehension, right? And Alan Turing wanted us to have comprehension about creating novel minds, to understand how it works. Now, what you may not know is that he was also very interested in morphogenesis, the idea of shape arising from the interaction of biological cells. Now, that's kind of curious. One might wonder why it is that a computer scientist would be interested in this problem. And I think that in keeping with how profound the rest of his work is, I think he understood something very clearly. And I think if he had lived longer, we probably would be further along in regenerative medicine, because he saw that these are fundamentally the same problem. And I think that actually is quite true. And I'm going to talk about that today. One important thing for us all to remember and to be humble about is that we all used to be single cells. So this is a lacrimaria. This is one cell. There's no brain. There's no nervous system. There's no cell-to-cell communication. This animal is doing things that if you're into soft robotics or engineering or anything else, we should make us very jealous. We have nothing remotely of this competency. And what it's able to do is handle all of its goals at the single cell level. So it's physiological, metabolic, morphological, behavioral goals, everything is handled. And during the process of embryonic development, we all make the journey across what's called the Cartesian cut. We start life as a single cell that many molecular biologists would see as a molecular network. It's what people call just physics. And then very slowly over the period of many months and then years, we transition into a creature that's going to make claims about its consciousness and its sort of first person perspective and so on. We all make this journey from physics to cognition. That's kind of amazing. I should say, although we don't have time to get into this now, that even at the lowest level, for example, the level of these gene regulatory networks, we found that there are, and other people have as well, at least 6 different kinds of memory learning, basically, that can be done in molecular networks. So actually, maybe there's not even a zero on this kind of continuum at all. Even at the beginning, there may be some cognition, but certainly we need to understand what kind of cognition is possible in what system. And one of the things that I've been working on is a framework that has at its core this idea of the axis of persuadability. So the idea is that with any system, be it evolved, designed, or combinations, and we'll talk about this, one gains practical advantage, meaning the most amount of control for the least amount of effort, by understanding where on the scale your system lies. And it may be something very simple that you can only deal with in terms of hardware rewiring. It may be something a little better, such as a thermostat that actually has an encoded goal state, and you can change the goal without having to rewire the machine. Maybe it's better than that, and you can provide rewards and punishments without knowing anything about how it works, which humans have done for thousands of years. Or maybe it's really complex, and in that case, you may communicate not even with training, but actually with reasons. And at some point, something like this, we're going to have ways to repair various artifacts that we make, and the best way to repair them is going to be to understand why they're upset and things like this. So this is a kind of scale. The idea is then that we can't assume where things lie on the scale. That's very important. We have to do experiments. And so the main points of what I'm going to tell you about today are basically these. All intelligence is collective intelligence. It's not just for ant colonies and bees and things like this. We are all made of parts. And in fact, biology uses a kind of multi-scale competency architecture, which I'll explain, as a set of nested problem solvers, each of which work in diverse problem spaces. I'm going to argue that the really useful invariant for recognizing, creating, and actually relating to unconventional agents is their degree of goal-directedness. And this leads to a very specific model of the cognitive boundary for scaling of mind from very simple agents to highly complex ones. I'm going to spend most of the time talking about anatomical control as an example. I'm going to show you an unconventional collective intelligence made of cells that operate in anatomical space or morphospace. And I'm going to tell you that the medium of this unconventional intelligence are electrical networks. They're a kind of proto-cognitive ancestor of brain function, and there are huge opportunities here for biomedicine, because we're going to be able to complement these kind of current hardware-focused approaches. And in the end, I'm going to talk about how synthetic bioengineering provides an incredibly large option space for new bodies and new minds that don't have standard evolutionary backstories. And so this raises many, many interesting questions for evolution, for medicine, and even ethics. So the goal of my framework is to be able to relate to truly diverse intelligences. The idea is that not only the kinds of things we're familiar with, but also really, really unusual creatures, colonial organisms, swarms, creatures that are made anew, that have never existed on Earth before, ones that may exist only in software or in some sort of engineering embodiment, and perhaps at some point off of the n=1 example on Earth, we may encounter life somewhere else. So we need to be able to understand what all of these things have in common, and we need to do it in a way that is not restrictive, but rather moves work forward, moves experiments forward towards new capabilities. So I'm going to do that in four parts. First, I want to remind us of some facts that we may not remember, and talk about the importance of thinking about continua instead of discrete kinds of creatures. And I'm going to introduce this notion of multi-scale competency. Then we're going to look at examples, specifically of morphogenesis as a collective intelligence and bioelectricity as a kind of cognitive glue that binds this holds it together. We'll come back then to some of the kind of theoretical aspects of the framework that we need to have to be able to explain how this all works. And then at the end, I'll introduce you to some novel organisms that have never existed on Earth before. So this is a famous painting of Adam naming the animals in the Garden of Eden. And this kind of view has been with us for a long time, because when people often think about these kinds of problems, they will talk about a human brain or a rat or some other kind of object as if this was a natural kind, a discrete category. And we're going to work to break that down a little bit. The first thing I want to point out is that cognitive subjects change during their lifetime. And so here you see a soft-bodied kind of creature that has to crawl around and chew leaves. It must become a flying, hard-bodied creature that drinks nectar. During this process, of course, the brain has to be different. So the brain during metamorphosis is basically liquefied. Most of the cells die, all the connections are broken. It rebuilds an entire new brain. So while people often think philosophically, what's it like to be a caterpillar? What's it like to be a butterfly? What's it like to be a caterpillar turning into a butterfly, right? This kind of severe remodeling makes the stresses of going through puberty as a child makes that seem just minor. This is an embodied mind changing drastically in real time. This is not just evolutionary timescales. So, and you can read about, and by the way, one of the most interesting things is that memories that a caterpillar acquires during its lifetime are still present in the butterfly. So despite the fact that, and you can read about examples of that here, despite the fact that the brain, the medium of this memory is taken apart and rebuilt, the memories remain. This is actually, goes even further in a creature we call planarian flatworms. So we'll hear much more about this today. These guys have the amazing property that they can be cut into pieces. The record is something like 276 pieces. Every piece knows exactly what a correct planarian looks like. They regenerate whatever is missing, and now you have multiple little worms. James McConnell in the 60s discovered this, and it was very controversial, but we've repeated it in our lab. It actually does work. You can train these animals on a particular memory. You can cut off their heads, including their brain. The tails will sit there doing nothing for about 8 days. They regrow a new brain. As they regrow a new brain and begin behavior, you can test them and discover that in fact they remember the original information. So what we have here is this fact that information moves through the body. We have this linkage between information of function, meaning learning and memory, tied very closely to information about structure, about the shape and the growth and form. And then, if you've taken philosophy 101 and talked about this kind of malfunctioning transporter, right, that instead of transporting you to another planet, it simply makes a copy and forgets to get rid of the original. And now you've got two identical individuals with the same memories. How many people are there? Planaria do this all day long, every day. So you can cut planaria, or in fact, they cut themselves into pieces. And then, now you have, all kinds of interesting questions about personal identity and so on. So what's interesting about biology is that of course, there are many levels of organization, so molecular, cellular, tissue, organ, and swarm, and so on. But it's not just structural. It's not just that we're made of parts that are made of parts. Every piece of every level in biology has competency, meaning that it solves problems in various spaces. That might be anatomical morphospace, it might be transcriptional space, the space of all possible gene expressions, space of physiological states, and of course, three-dimensional behavior. And what evolution does is use some of the same tricks in all these different spaces, and all of these different levels cooperate and compete with each other to accomplish their various goals. So this becomes very important. So here's a simple example. This is a tadpole of the frog, Xenopus laevis. Here is the brain. Here are some nostrils. The gut is down here, and this is the tail. Now you're going to notice something very strange about this tadpole. Here's where you would expect the eyes to be. There are no eyes here. We've engineered it to instead have an eye on its tail. Okay. Now, this animal for a very long time has evolved for the brain to expect visual input from two specific locations into the optic tectum. We can create these animals. It turns out they can see perfectly well. So we built this machine to train them for visual learning. So things like stay out of a moving blue spot and things like that. They can see quite well. So what you see here is this incredible competency of the eye primordium. It finds itself sitting next to muscle instead of brain. No problem. It still knows how to make an eye. Then the optic nerve comes out. It needs to connect somewhere. Oftentimes it'll find the spinal cord and connect to the spinal cord, and the animal can still see. So you can see what an incredible feature for evolutionary evolutionary change and efficiency it is when you can depend on your parts to do their job even when things change. So this goal directedness and competency of the lower levels buffers the negative consequence of mutations. It means you can explore all kinds of things because as your organs move around and things change, they can still perhaps get their job done, right? So this is very important. Now, as we think about these parts, we realize now that because of chimeric technologies and bioengineering, all of the parts are swappable. So all the way from molecular components all the way up to the creatures of a hive colony, you can swap any piece of this out for other objects, whether it be evolved material or something engineered like a nanomaterial or a smart chip or something like that. It's somewhere along this scale, right? It could be evolved, it could be designed or some mix thereof. It could be a very simple thing, passive. It could be a very intelligent machine. You can now swap all these things. And so now we realize that you really can't rely on old categories in terms of what something looks like and what part of the evolutionary tree it comes from to understand or estimate their intelligence or their agency. We already have all kinds of cyborgs and hybrids and all sorts of combinations that never existed before. And so in this idea that there's this massive tree of possible living beings, we need to find an invariant. We need to find something that they all have in common, something that we can use as a parameter to organize them, to compare them with each other, regardless of what they're made of or how they got here. Now, this was already back in the 1940s. Rosenbluth, Wiener, and Bigelow were already thinking about this. They wrote a really nice paper where they proposed this idea, the scale for all of these creatures, whether they be machines, the artificial machines or natural machines, evolved organisms, they would be somewhere on this scale where the invariant is goal directedness in the cybernetic sense, not in a mystical, sort of sense, but in the cybernetic notion. Okay, and so what I want to do now is go over some examples with you about what this means in biology. Okay, what goal directedness looks like, and in order to do that, we have to ask some very basic questions that a lot of times we gloss over in teaching developmental biology and things like that. Where do anatomies come from? So this is a cross section of a human torso. You can see this remarkable complexity, in fact, invariant complexity in all normal humans. All the different organs, tissues, structures, everything is in the right place, the right orientation relative to each other. Simply amazing. And of course, it starts off as this. It's a ball of early embryonic blastomeres. Somehow it has to do this. Where is this information? Okay, how do they know what to build? Now, you might be tempted to say DNA in the genome, but we can read genomes now, and we know that none of this is directly in the genome. Genomes specify proteins. They specify the micro hardware of every cell. So lots of questions. How do cells know what to build and when to stop? If a part of this is missing or damaged, what actually do we need to do to convince these cells to rebuild exactly what was there? They did it once. Why can't they do it again? And as engineers, and we'll get back to this at the end of the talk, how far can we push this? Could we get the exact same cells to build something completely different than their genomic defaults? Okay, and so we need to really understand this. So you can imagine why this problem is so important, because let's think about the end game of medicine. What do you want in the end? You want something that we can call the anatomical compiler, meaning that if somebody should be able to sit down in front of the computer and draw the anatomy of the animal or plant that we want, not the molecular pathways, but actually what we care about, which is the anatomy, right? So you draw this thing, and if we knew what we were doing, the software would compile that down into a set of stimuli that would have to be given to individual cells to get them to build whatever this is. Why do we want such a thing? Because with the exception of infectious disease, all problems in biomedicine, so birth defects, traumatic injury, cancer, aging, degenerative disease, all of this would be completely solved if we knew how to tell cells what to build. Okay, and so this is central to all of medicine, and yet we are nowhere near being able to do this. Why is that? We've had so much progress in genomics and stem cell biology. Why can we not build this? Well, let's ask a simple question. Here's a baby axolotl, a salamander. They have legs. Okay, frog larvae do not have legs, so tadpoles don't have legs. We're in the process of making a frogolotl. Okay, this is real. These things are quite viable. So you take some cells from an axolotl embryo, combine them with cells from a frog embryo, and you've got this chimeric frogolotl embryo. Now, you have the axolotl genome, you have the frog genome. A simple question: are frogolotls going to have legs or not? And if they do have legs, will those legs be made only of axolotl cells or of frog cells? We have no models in biology that can give us an answer to this other than try it and see what happens, okay? Even though you have full access to the genomes. And in fact, it's actually even worse. Even without making these chimeras, just from being given a genome, you have no idea what the size, shape, symmetry type of the organism is going to be. You can compare it with other genomes that you already know, but that's cheating. So the idea is that we understand quite a bit about the mechanisms that are required for cells to accomplish certain things. So all these molecular pathways, we're really good at this. We're a very long way away from control of large-scale form and function. And in particular, what we need to understand is how do cellular collectives know what to do, in particular in novel circumstances. And I'm going to suggest that's a kind of intelligence. And modern molecular medicine has been operating largely at this level. Everything today is very exciting with respect to stem cell biology, genomic editing, CRISPR, protein pathways, protein engineering. All of the focus is on the molecular hardware of life. We still really do not understand the software, and yet we know how much computer science and information technology benefited when we moved from programming computers this way to exploiting software properties. So I want to talk a little bit about intelligence, and William James gave a really interesting definition of intelligence, which is the ability to reach the same goal by different means. And there are two interesting things here. One is that it focuses on goal-directed activity, and I think that emphasis is exactly right. And it's this, it emphasizes this flexibility. The idea is that in proportion to how intelligent we can say that a system is, it has the ability to overcome novelty, perturbations, things like that, to still reach their goals. So I'm going to show you some examples. I told you that embryonic development is very reliable, which is fantastic, but it isn't hardwired. So if I take an early mammalian embryo, I cut it in half, I don't get two half embryos, which is what would happen if you cut any of our technology in half. You get perfectly normal monozygotic twins. Each side knows what's missing, rebuilds. Now you got two creatures. Amazing. It gets even more interesting. This is the lumen of a kidney tubule in a newt. And normally they're made of, let's say 8 to 10 cells working together to make this structure. One thing you can do is force the cells to be bigger and bigger, okay? And if you make bigger cells, the animal remains the same size. It's just that fewer cells now participate in this. That's already interesting. It's an adjustment. Here's the real kicker. If you make the cells gigantic, just one cell will bend around itself and give you the exact same lumen. Now, what's interesting about that, and this is not our work, this has been known since the 40s. What's interesting about that is that this is a completely different molecular mechanism. This is cytoskeletal bending. This is cell-to-cell communication. So different molecular components are being called up in an entirely novel situation to implement the same high-level anatomical goal state. Okay, this is the ability to get to the same goal despite perturbations, not only perturbations of the environment, such as injury, but actually changes in the parts that you have to work with. We don't have anything remotely able to do something like this. Okay, now, in addition to development, there's a more general capacity, which is regeneration. So this is an axolotl. These guys regenerate their limbs, their eyes, their jaws, their spinal cords, portions of the brain and heart. And if you cut off the limb, you can cut it at any level. It will grow exactly what it needs to be a perfectly correct salamander arm. So you can see that now it gets to the same goal from different starting positions. And the most amazing thing about this process is that it stops. When does it stop? It stops when the correct salamander target morphology for that arm has been complete. So that's the stop condition for this computation. It actually has to know what a correct salamander arm is. By the way, regeneration is not just for so-called lower animals. The human liver regenerates. Even the ancient Greeks knew that. How they knew that, I have no idea. It's actually a very interesting question. Deer regenerate their antlers every year, up to 1.5 centimeters of new growth per day when this bone and nerve and everything else is growing. And in fact, human children regenerate their fingertips below a certain age. If they have a clean amputation without sewing over the skin, they'll just regenerate. Okay, so to continue this theme of same goal by different means, this is the collective intelligence of cellular groups working out some interesting problems. One of the things that they can do is reach the same goal in novel paths through morphospace. So this is a frog. This is a frog leg. Frogs, unlike salamanders, do not normally regenerate their leg, but we can make them do that. I'll talk more about that momentarily. If we induce that, what you see here is that the legs regenerate. Eventually, you get a very nice looking leg. However, the intermediate steps don't look anything like the developmental path that limbs take in order to form. In fact, they look much more like a plant. You've got the central stalk, you've got these toes coming off the sides, you got a toenail, and yet they come to the same spot afterwards. So this collective intelligence is able to navigate morphospace in a new path, and this really takes off in this example where we discovered this a couple of years ago. This is a tadpole, of course. Here are the eyes, the brain, the mouth. In order to become a frog, tadpoles have to rearrange their face. So the jaws have to come out, the eyes have to move forward, everything has to move. And it was thought, prior to this, it was thought that what the genome specifies is a kind of machine that has every part moves in the right direction, the right amount, and you go from a standard tadpole to a standard frog. So what we did was, so we wanted to look for some intelligence in this process. What we did was we made so-called Picasso tadpoles. Everything is in the wrong place. The mouth is off to the side, the eyes on top of the head, everything is off to the, everything's in the wrong place. Amazingly, they give rise to pretty normal frogs because all of this stuff is going to move around in novel, unnatural paths. In fact, sometimes they go too far and have to come back a little bit. And then it all stops when they reach a correct frog face. So what the genetics actually gives you is a machine that is able to execute an error minimization scheme. Okay, it's able to reduce the delta between whatever the shape is now and the correct state, and it will do so despite various the unusual starting conditions and so on. So that raises all kinds of interesting questions. So in my group, what we've been doing is this here is the sort of typical paradigm view of developmental biology, that it's a feed-forward, open-loop kind of process. That what you have here is gene regulatory networks. The genes will turn each other on and off. There will be some proteins that do things. They're sticky, or they diffuse, or they do exert force, or they do something. And this highly parallel physics will, through the process, this magical process of emergence, when you have lots of very simple rules being followed in high, in parallel in high numbers, there will be an emergent complexity, symmetry breaking, all of that, and then you get this wonderful outcome. And all of that is true. All of that does happen, but it's not the complete story, because we now know that when you deviate the system from its correct target morphology with injury, with mutations, with teratogens, feedback loops will kick in, either at the level of physics or genetics, that will try to get back to where it's supposed to be. Now, of course, feedback loops are nothing new in biology. Everybody understands feedback loops, but there's a couple of interesting twists on it here. The first thing is that, and so by the way, what this is it's a basic homeostatic cycle. It's pattern homeostasis, right? Like you have in your thermostat. The twists are severalfold. One is that we understand feedback loops, but we usually think of single numbers, scalars, like pH, temperature, hunger level, right? In this case, the set point for this process is a fairly complex state. It's a coarse grain representation of geometry. So that's kind of interesting and unusual. The other unusual thing about it is that we are not used to thinking about biology working towards goals. You're supposed to sort of try to right? We're all taught to try to reframe our explanations to be kind of emergent from low-level rules. But of course, cybernetics and control theory give us ways to think about this that isn't magical. So that's okay. So having this hypothesis, it makes a very strong prediction. The prediction is this. If this is true, if the animal has in some way a represented set point, the way that all homeostatic systems have to, then if we were able to find that set point, meaning how is it encoded, we were able to decode it, and we were able to rewrite it, then we should be able to overcome an important limitation in medicine. The problem here is this: if you want to make changes here at the system level, and you believe that everything percolates up from this level, you've got a terrible inverse problem to solve. How do we make changes down here to have specific outcomes here? With the exception of low-hanging fruit like single gene diseases, it's entirely likely that CRISPR technologies, genome editing, all of that is going to reach a ceiling because it's completely unknown what it is that you would change here to modify the shape of somebody's limb, to give them back an eye, these kinds of things. If this is true, then you can do what you do with a thermostat, which is that you can change the set point. You don't have to rewire the thermostat. In fact, you don't even need to know how the whole thing works. What you do need to know is where the set point is and how to change it. So could this really be true? And we started thinking about this years ago. And for inspiration, we turned to the first system the one system that very obviously in biology, uncontroversially, stores goals and pursues them, and that is the brain. So in the brain, the system works roughly like this. You have some hardware. The hardware consists of a network of cells every cell has these little ion channels. They send ions in and out and basically allow a voltage potential to form across the membrane. And that voltage potential may or may not propagate to their neighbors through these electrical synapses known as gap junctions, so selectively. So that's the hardware. And the commitment of neuroscience is this idea that the electrical activity going on in this network, and you're seeing this is a lovely movie that someone made of a zebrafish brain. This is electrical activity in a living zebrafish brain. The idea is that that is the medium in which the cognitive content of that mind is stored, whether it be a human or a zebrafish. If we knew how to decode it, you would have access to the memories, the preferences, the goals, everything else that belongs to this mind, right? So by reading the electrical pattern, you literally have access to the information content of that mind from a cognitive perspective. Now, what's interesting is actually that every cell in the body does this. So it turns out that every cell in your body has ion channels. Most of them have gap junctions with their neighbors. They all make electrical networks. And we can do something very similar to the idea of neural decoding in neuroscience. And we can try to decode what it is that the rest of your body tissues are thinking about. They're not thinking about three-dimensional behavior. I'll show you in a minute what they are thinking about. This is a companion to this video. This is not a model. This is a real data. This is a voltage-sensitive fluorescent dye. This is a movie of an early frog embryo created by my colleague, Danny Adams, where you can actually see where all the electrical states are as these cells try to figure out who's going to be anterior, posterior, left, right, and so on. And so the parallelism between the nervous system and electrical activity outside, which we call developmental bioelectricity, is extremely strong. So whereas the electrical activity of your brain is used to control your muscles, to move your body through three-dimensional space, the exact same kind of activity, only slower, of the rest of your body networks are used to control your cells to move the configuration of your body in morphous space. Okay, everything else holds. It's a very similar system. Now, we shouldn't be surprised about this, because if we ask where did neurons come from, we find out that, of course, they didn't just sort of appear out of nowhere. They speed optimized things that pre-neural cells have been doing for a really long time before brains and nervous systems came on the scene. Evolution figured out that bioelectricity was a great way to process information around the time of bacterial biofilms. It's an ancient thing, and you can read more about that. So developmental bioelectricity is the precursor to the kind of cognition that people study in neuroscience. Now, what we did was we created some tools to allow us to read and write the thoughts of the body, basically the information content that was present in somatic networks that we were testing this hypothesis that it had something to do with controlling morphogenesis. So first we applied this dye technology that just allows us to take measurements and see where all the electrical states are as a function of time. And so this is a time-lapse video. We do a lot of computational modeling to understand, given the various channel and pump genes, why do they give rise to these specific patterns? And I'm going to show you some interesting patterns. This, for example, is an early frog embryo putting its face together, again, time lapse. And so you'll see here, this is grayscale, the same voltage dye, all kinds of things going on. Here's one frame out of that movie. And I'm showing you this example among many others because here's the most obvious. We can see this in the easiest way, which, neuroscientists should be jealous because patterns in the brain are not this easy to decode. And of course, most of our patterns are not either. This is an example that happens to be very straightforward. You can actually see where everything is going to be before the genes turn on to regionalize this ectoderm to the face. Here's where the mouth is going to be. Here's where the animal's right eye, the left eye actually comes in later. Here where the placodes are. All of this, you can already see a representation of what an embryonic face looks like before any of that happens. Now we know that this pattern is instructive, because if I modify this bioelectrical pattern, I'm going to tell you in a minute how we do it, then I can reproduce, or I can move all these organs and reproduce them elsewhere. That's how we make the Picasso tadpoles. It's by messing around with this. We call this the electric face. And so by messing around with this instructive pattern, you can change where everything forms. That is a natural pattern that is absolutely required for normal development. In fact, human patients with channelopathies will have craniofacial defects and other defects exactly for this reason, because it screws up the electric phase. Now, here's a pathological pattern, which we'll come back to shortly, having to do with cancer. So if you inject an oncogene, let's say for a nasty human KRAS mutation into this animal, eventually it makes a tumor. But before these tumors become apparent, already you can see the bioelectrical consequence of this oncogene. It basically decouples these cells from the network, and they will transition to a unicellular-like existence, treating the rest of the animal as just external environment, and sort of go off and do amoeba things that amoebas like to do. So this is a way to track these. Now, much more important is the ability to write the information, to do functional experiments. Now, I want to be clear, this is all of the phenotypes I'm going to show you today. Nothing about electrodes or applying electrical fields. There's no electromagnetics, so there's no waves. Everything is molecular physiology. So the idea is, in any kind of tissue, we really have only two things we can do. We can control which cell talks to which other cell. So we can control the topology of this network. And we can do that by mutating these gap junctions, by opening them and closing them with drugs, that kind of thing. Or we can go in and use either optogenetics or drugs, or again, mutating channels that will control the electrical state of the individual cells. So we can write the information. And of course, the latest technology is optogenetics. So we can actually put down light masks onto these animals, onto these tissues rather, and really control what all the electrical patterns are going to be. Okay, so now what happens if you do this, right? What is actually the evidence? And of course, when I first suggested that we were going to do this, now back in 1999 or so, the basic assumption was, and was what everybody said, is that voltage is a housekeeping parameter. If you mess around with cell voltage, you're going to get uninterpretable death, and that's all that that there's going to be to it. So I'm going to show you some things that actually happen, because it is not a housekeeping parameter. It is an informational medium. Now, one example is if I take this early frog embryo and I take some cells that are going to become gut here, and I inject RNA encoding specific potassium channels. We choose these channels because we know, we can calculate that they're going to reproduce an eye-specific pattern in whatever cells they landed. When you do that, guess what the cells end up making? Even though they're endodermal cells that normally would be fated to be gut, they'll make an eye. This, by the way, this eye can be formed anywhere on the embryo. The so-called master eye gene, PAX6, does not do that. It only forms eyes up here in the anterior neuroectoderm. This way you can make eyes anywhere. They have all the right layers, optic nerve, lens, retina, all of that stuff. Notice two interesting things. One is that this is modular at the level of organs. We did not give enough information to specify how to make an eye, for good reason. We have no idea how to make an eye. It has all kinds of different cell types. We don't know how to micromanage their construction to, and to control all the gene expression. We don't know how to do that. What we did do was find a trigger, a subroutine call that basically kickstarts the eye building program. And we just said, here, build it right here. You already know how to do it. Go ahead. And the whole thing is self-limiting. So it didn't make a giant eye that takes over the whole embryo. It doesn't make a tumor. It makes an exact eye. So that's the first thing. It's highly modular, which is very good for regenerative medicine. That's great. So you can call up simple triggers, right? We'll get back to the issue of memory momentarily. Simple triggers give rise to very complex recall of action. Now, the other thing is, this is a cross-section of a lens sitting out in the tail of a tadpole somewhere that we induced. The blue cells tagged with this beta-gal enzyme are the cells that are actually carrying our new ion channel. But there's not enough of them to make a proper lens. So while we are instructing these cells to make a lens, they do a secondary instruction and they tell their neighbors that, hey, all of you need to work with us to help us complete this lens. These cells down here were not themselves affected by us at all. So there's two levels of instruction. We instruct these cells instruct their neighbors, and they complete this very specific instruction process, even though we only managed to inject half of them. Okay, so that's very interesting. There's all kinds of modular instruction going on here, not just by us. Now, in addition to eyes, what else can you make? Well, we can make extra forebrains. We can make ectopic otocysts, which are balancing organs. We can make ectopic hearts, as you can see here. We can make legs. So this is, we have 5- and 6-legged frogs. You can see that. And something else that's interesting, you can make fins. Now that's a little odd because tadpoles shouldn't have fins, and we'll get to that. We'll get to that momentarily. But so we can call up, we can call up all these different organs. Many other organs we have no idea how to make yet. We're still in the process of cracking this code. But of course, it has lots of biomedical implications, for example, in regeneration. So frogs, unlike salamanders, you amputate the leg, 45 days later, there's nothing. What we did was design an ionophore cocktail that would put the wound in a pro-regenerative state. When you do that, it kickstarts things like MSX1, which are regeneration genes. And eventually you get a leg. The leg is touch sensitive, it's motile, as you see here, and eventually a pretty respectable leg. We are now in the process of doing this in mammals, ideally, hopefully someday in human patients. We're nowhere close to humans yet. We're just working this out. I have to do a disclosure here. This is David Kaplan and I are co-founders of a company called Morphoceuticals Inc., which they make these bioreactors that are wearable delivery vehicles for the signals that we're trying to give. And the important thing about this approach is that we are not going to 3D print or micromanage at the level of stem cells this process at all. In the frog, you put the bioreactor on for 24 hours, you get 18 months of leg growth. During that time, you don't touch it at all. The point is not to control that process throughout. The point is to change the decision-making of those cells immediately after the injury, where they're trying to decide, are we going to scar, are we going to build something? At that point, you change you shift the circuit, and then you don't touch it again. That's our ideal goal. Okay, so I want to switch back to these planarians. Planaria are very interesting. They have a true brain, most of the same neurotransmitters that you and I have, lots of internal organs. Of course, they're regenerative. They also are immortal. That's interesting. There's no such thing as an old planarian. They do not age at the level of the organism. Any cell that senesces and dies, they regenerate it. That tells you something that these ideas about thermodynamic limits on lifespan are in fact not real. Also, they don't have multiple copies of p53 the way that people think elephants need to keep away from cancer. They do not, planaria don't get cancer. They're incredibly regenerative. It's because of their incredibly tight control over their anatomy. They have something like 30% of this animal is plastic stem cells, and yet there's no cancer here. So what I want to use them for is something very interesting. Here's a planarian. It's got one head, one tail. The anterior gene marker is expressed in the head, like it should be, not in the tail. We amputate the head, we amputate the tail. This middle fragment will regenerate this normal worm 100% of the time. It's got incredibly high fidelity. But here's something has happened. Again, one-headed worm, head and tail. The gene expression is normal, but when we cut him, we got a two-headed worm. This isn't Photoshop; these are real animals. But I just told you it's a very robust process. Why is this happening? Well, because we found an electric circuit that actually tells these animals how many heads they're supposed to have. And what we did in the meantime was here's the pattern, here's the information stored in this electric circuit that says one head, one tail. What we did was, and you can see it's kind of messy, the technology is still being worked out. What you see is that we basically incepted a false memory into this collective. We said, no, actually a planarian should have two heads like this. And sure enough, two heads they make. Now, here's the most important thing about this whole story. This is not a map of this two-headed animal. Okay, the bioelectrics is not a readout of what the anatomy is doing now. This is a map of this one-headed animal. So what you're seeing here is that a normal one-headed anatomy can store, it can represent at least one of two memories or representations of what a correct planarian is supposed to look like. Now, until they get injured, they ignore it, and so it's kind of a latent memory. But as soon as they're injured, this is what guides the cells when we ask the question, how do they know what to do and when to stop? This is it. That when I showed you that feedback loop and I said that a homeostatic circuit has to store a set point somewhere, we are now looking at it. You are seeing the encoding of the set point, and not only are we seeing the set point, we know it's instructive functionally because we can change it and the anatomy follows. And probably there are many other things it can store, but this is the one we've nailed down the best. And by the way, this may be, if you think about where complex cognitive capacities come from, this may be the origin of counterfactual thought, right? This is a simple circuit that can think about what it's going to do if it gets injured at a future time. This is information that is not at all related to what's going on right now, and it's not related to what all of its lengthy past history was. It's about the future. Okay. Now I keep calling this thing a memory. Why do I do that? Because if I take one of these two-headed animals and I chop off the endogenous head, I chop off this crazy secondary head. You know, people will say, well, the posterior tissues were epigenetically reprogrammed. Okay, fine. We throw them in the garbage. We just keep the nice normal middle fragment. There's no genomic editing here. The genome is wild type. So you would think the standard paradigm would be that, well, what else could they do but regenerate normal animals? All of their genetic instructions are for how to build a one-headed animal, right? Wrong, because these middle fragments will continue to build two-headed worms forever, as far as we can tell, until we come back, we actually know how to reset them back to being one-headed. So there's a few interesting things here. This is a memory because it's long-term stable. It's rewritable now that we know how to do it. It has latency or conditional recall. There are discrete behaviors that it can remember in one-headed versus two. And so because the genome is completely wild type here, the answer to the question of where is the determinant of the number of heads in a planarian is not the genome. What the genome does is gives you some hardware whose default behavior says build one head. That's just the default behavior. The hardware is actually much more interesting than that. It stores that information in a stable electrical state of a circuit, like a flip-flop, like a volatile RAM kind of thing. And you can come along and change it. And when you do change it, it will stay. It remembers the changed state. You can go back and forth. You can change it. So the answer to how many heads you have is interesting. It's the memory that you have in that electrical circuit. And what the genome does is set the default state. Now here's a video. This is what these two-headed animals do in their spare time. I always show this video now. The first time I presented these data at a meeting, someone stood up and said that's impossible. Those animals can't exist. And so now I make sure there's a video so everyone can see. These are from multiple cuts, right? And what we're doing now is trying really hard to merge together the two lenses through which people see this stuff. On the one hand, it's an electrical circuit and it's a piece of physics, and you want to understand the stable attractors and, you know, why does it do certain things in certain states. But on the other hand, we have this really interesting connectionist paradigm that tells us how networks remember things. And in particular, electrical networks that can do pattern completion, right? So machine learning is all over this stuff. When you can make networks that learn such that when a piece of the pattern is missing, the network will give it back to you. And there's of course also a kind of a thermodynamic view on this. So we're trying to merge all this stuff. Now I've told you that you can make worms with two heads, but you can go even further than that. You can actually find attractors in this circuit space that belong to other species. So if I take a planarian with a triangular head, and we amputate the head, then what this and then for about 48 hours we use a drug to prevent the cells from electrically talking to each other. So we basically fragment the network. It stays in this really weird fragmented state. We pull out the drug, everything settles down to a stable state. When that happens, sometimes this is a stochastic process. Sometimes it comes back to the correct triangular head. Other times it makes a round head like this like this S. mediterranean. Sometimes it makes a flat head like this P. felina. The frequency of these heads is proportional to the evolutionary distance from here to these other species, somewhere between 100 and 150 million years distance. The genome hasn't changed. It's the same exact hardware making completely different types of heads, not only the shape of the head, but the shapes of the brain, the distribution of the stem cells, because it can find these other attractors in the state space that other species use by default. Okay, so you can knock it into these other attractors. You can actually go even further than that, and you can find regions of morphospace that evolution does not use because they're not ecologically competitive. You can make these crazy spiky forms. You can change the symmetry type and make it a cylinder. You can make a combination that has a flat, flatworm with a tube going into the third dimension, right? And so what we're doing now, and this is the key to biomedicine, is to make a full stack multi-scale set of models where we start with the genetic description of what are the channels and pumps that exist in these cells, so transcriptional networks. Then we simulate the physiology that results from this in the tissue, that is, where do the ions go and what are the voltage patterns that result. And that's built up into large-scale body-wide patterns and the rules, and we extract the rules by which they change. And ultimately what we want is these kind of algorithmic models that basically enable us to, without having to solve the inverse problem, enable us to make rational changes. If we understand what the logic is, right, we can make certain changes. So we want to go from the components, from the physics, all the way up through the physiology, and ultimately to something that looks like decision making. And the way that works is, and this is our collaborator Alexis Pitek made this incredible simulator, where you can, it's a virtual, you can make a virtual creature. Every cell runs whatever circuit of ion channels and genes and whatever else you want to put in there. And then you can ask system level questions like, hey, when I cut this in half, how come it rescales? The electrical pattern rescales, or maybe it doesn't rescale. So you can do all that. Now, one of the things you can do with this model, and this is just one of many examples, is you can make very targeted repairs of complex defects. So here is a frog, a tadpole brain. Okay, here's the forebrain, midbrain, and hindbrain. And what you can do is, let's say you expose them to some kind of teratogen, alcohol, nicotine, mutation of neurogenesis genes. What you will see is that the normal bioelectrical pre-pattern that controls where the edges of the brain should go can go awry. And what we did was we made a computer model, and this is also the work of Alexis Pitek and a staff scientist in our group, Vaibhav Pai. They made a computational model of what actually goes wrong in these cases. And then they were able to ask the model, well, given that the bioelectric pattern is now wrong, in other words, you're unable to remember the correct shape that you're trying to make, what channels would we open and close to get the shape back to normal? Okay, so you can interrogate that model. That model made a prediction. There's this one interesting ion channel called HCN2, which would actually, and this is not at all obvious, it's such a nonlinear thing, you can't just guess. So you have to do the simulations. But it told us that there was an interesting channel that would actually fix the pattern. So we tried it. And to my great amazement, when models actually make predictions that turned out to be correct, what happens is, so here again is the normal tadpole brain. Now here, what we did something even worse than teratogens. We mutated Notch. Notch is a really critical neurogenesis gene. Okay, it's required for normal neural development. When you mutate Notch, the forebrain is gone. The midbrain and hindbrain are a bubble. These animals have no behavior. They just lay there, just terrible brain defect. On this background, we showed two things. You can either introduce new HCN2 channels, or you can just use drugs. In fact, already human-approved drugs to open the native HCN2 channels. And when you do that, you get your normal brain back. You get the brain structure back. In fact, these animals have IQs in the learning assay that are indistinguishable from wild types. So now, this is where this field has gone. When we started, we were doing screens of bioelectric modifications to find out what is the space of possible things that bioelectricity controls. Now we're to the point where we have an actual computational model of the circuit that can predict drugs. So this is not, that's very important to realize that we're now, and again, this is still very early, this is baby steps compared to what's possible. But basically we're to the point where we can understand these circuits and make models that allow us to infer interventions. And so the idea is, and some, there's a simple version of it up that people can play with, and there's some software that you can download. But the idea is that someday you're going to be able to sit in front of the software, you're going to pick some tissues. The software will already know from RNA-seq studies what all the control knobs in this tissue are, meaning all the channels that are expressed, and it will know what the correct pattern is, and it will then simply help you pick drugs that open and close the correct channels using that simulator. That, of course, is the key part, the bioelectric simulator. So lots and lots of work to do, but we can already see the shape of this tool coming. So now I want to back off from the biomedical stuff, which is important because I'm going to give you a bunch of ideas that border on philosophy, and it's really important to know that this stuff actually drives experimental progress, right? It's not just the stuff that you can sort of take it or leave it the way that a lot of philosophical claims are. No, this actually is testable, and it's driven progress. So let's just look at some of this. One of the things we know is that goals can change scale during the lifetime of an individual. So individual cells have very, very simple goals. They can scale up, they can merge together to pursue very large scale goals. For example, the goal of maintaining a limb. Now, individual cells have no idea what a limb is. Only the cellular collective can measure these states to know if the limb is right or not. In fact, many of them will have to die. All the stuff in between the fingers has to die. So this is a goal that the individual cells aren't following it. The collective is following it. So the goal has grown, but it can also shrink. And so this is glioblastoma in culture. And so what you can see here is that these cells have become electrically isolated from the network. They go back to their primitive unicellular lifestyle where, what are their goals now? Well, every cell wants to become two cells, and that famous quote, and to migrate to where life is good. So now metastasis, transformation and so on. Okay, so the goal now what's important to think about is that a lot of times people think of cancer cells as being more selfish than normal cells. I actually think that's wrong. I think they're exactly as selfish as any other biological creature. It's just that their cells are tiny. So these cells are all being very selfish as well, but their self is huge. Their goal, and they're taking energy away from other organs and so on. Their goal is to make this thing. So their self is rather large. The self can shrink. The self can grow and shrink. And so what that means is that we can start to think about cognition as the size of the goals that individual cells are capable of pursuing. So a single cell may have a little bit of memory going backwards, a little bit of anticipation capacity going forwards, but a network of cells can measure much larger areas. They can have more memory and more anticipation because they have more computational hardware, basically. This is, again, this is just to say, this is not just empty theory. We actually use this. This is that tumor that I showed you. We said, okay, what we're going to do is we're going to, along with that oncogene, we're going to inject an ion channel that's going to force you to remain connected to the electrical network. Meaning, yes, you've got the oncogene, which theory says should transform you, except that you're still going to be electrically connected to the network that remembers what a correct set of organs and tissues are. And this is what happens, right? The oncogene is labeled in red. You can see there's tons of it. It's blazingly strongly expressed. In fact, it's everywhere. There's no tumor. And there's no tumor because this cell is not being allowed to go off on its own. It's being forced to be part of this larger scale tissue level system that is basically not into making tumors. Its goal state is maintaining nerve and muscle and everything else that's there. Okay. So what you can then do is you can think about all sorts of creatures. In fact, any arbitrary creature, be it a natural creature, an evolved, an alien, whatever you want. And you can plot it on a diagram that tracks the scale of the biggest goals that creature can pursue. So this kind of looks a little bit like those Minkowski relativity diagrams where I've taken all of space and put it on one sort of axis, time is here. And so what I simply ask for any kind of creature you want to produce, I'll say, okay, what is the largest in terms of space and time goal that it's capable of following? Is it just measuring local butyrate concentration and it's got a memory of a few seconds and so on? Maybe it's a bacteria or a tick. Does it have a memory backwards of some number of months and anticipation forward a little bit? But it really can't care about what's going to happen 3 months from now and 10 miles over. It's just impossible, right? If you're a human, well, you might be working towards world peace, and you might be stressed out by the financial markets, and you might be depressed that the sun is going to burn out in some number of billion of years. Your goal, your ability to represent goals are enormous, right? It's huge. And so we can define a cognitive horizon, almost like a light cone of things that you can possibly care about. Humans maybe uniquely are capable of caring about goals that are bigger than their own lifespan. And so that probably has a lot of psychological ramifications for us that other creatures don't have to deal with, because if your goals are small, they are guaranteed to be achievable in your lifespan. We are maybe uniquely can have goals that are definitely not achievable in our lifespan, right? So we can think about that. But the idea is that you can compare all of these different types of cognitive systems on the same kind of graph, no matter what they're made of or how they got here. It's the central invariant is what kind of goals can they follow. And in fact, we can make some very specific models about how these goals scale up. So to go from here to here, from a single cell that has this very small homeostatic loop of things that it measures, compares, and then acts. Once you start connecting cells with gap junctions, with these electrical synapses, everything scales up. What every cell measures is in part a function of what its neighbors measure. So now they're measuring something very large. They're in all of these pieces, the set points become bigger, the actions become bigger, and so their goals scale up. Okay, and so this is one way to scale them. One of the important aspects of how they scale is the fact that gap junctions have an interesting property. The gap junctions are kind of magic because, unlike regular signaling. See, when this cell signals to this other cell through, let's say, a secreted molecule, this cell knows perfectly well that the signal came from outside. And so it can ignore it. It can choose to do something about it. But the boundary between it and another cell, the individual identity is very strong, right? It's very strongly maintained. What happens with gap junctions is that they provide access from each cell to the intracellular milieu of the other cell. So that means that when this cell experiences something, right? So something happens and there's a calcium spike, and then, you know, so that's the engram of that memory of that event, right? The cell has a memory. That calcium can propagate to the neighboring cell. It has no metadata on it about who it belongs to. And so this cell now receives that same calcium. It has no idea whether that's my memory or whether that's your memory. So what these things do, in effect, what the gap junctions do, is they dissolve, or at least partially, the boundaries between individuals. It's like a mind meld, because the information now becomes shared. Once that happens, you are automatically growing in cognition, because not only do your goals get bigger, it's impossible not to cooperate. You can't, you know, in the game theory sense, you can't defect against yourself. You're basically, whatever you do to your neighbor, it comes back to you immediately. You're linked. So it erases. It's kind of interesting. So my memory becomes our memories, my stress, whatever you're stressed about, becomes our stress, and my goal becomes our goal. And all of these things become larger. So in the last couple of minutes, I just have a few minutes, what I want to do is show you these novel organisms, because we've been talking a lot about goals and the idea of goal directedness as a central aspect of scaling of cognition. We can ask interesting questions. Where do these goals come from? Right? And the standard answer, of course, is evolution and selection. So we started this project a few years ago. This was driven in my group by staff scientist Doug Blackiston, and then we worked very closely with Josh Bongard at University of Vermont, and Sam Kriegman, who was his grad student at the time. And this is part of this new Institute for Computationally Designed Organisms. We asked a couple of questions. The first question is, how much plasticity is there in cells building things? Of course, there's the genomic default, you know, fish eggs make fish and acorns make oak trees, but how much plasticity is there? And then the question is, what do cells actually want? Because we don't see the cell. So let's look at skin. Skin has this boring two-dimensional life on the outside of an embryo. It keeps out the bacteria. We don't know what the skin cells want, because normally they're under instruction from all the other cells. What happens if you give them a chance to kind of reboot their multicellularity? What are the backgrounds? And this is important, because evolution doesn't work on a blank canvas. Evolution has to modify the agendas of cells, much like we, as I'll show you in a minute. Evolution works with this kind of agential material, and so it has to deal with whatever it is that cells are going to do, and then modify that in various ways to improve selection. So here's what happens. This is a frog embryo at about 12 to 18 hours. You take the animal cap cells. These are fated to be skin. You dissociate them into here. You can see that they're being pipetted. So this is a dissociated clump of cells. They're being pipetted into this little hole. Now, there's many things they could have done. They could have died. They could have crawled off. They could have become a nice flat monolayer, the way cell cultures do. They didn't do any of those things. They combined together like this. They sort of condensed, and then what you're seeing, the flashing, that's actually calcium signaling. So already we can tell something interesting is going on. It looks very, very brain-like. And then what they do is they become these mobile little creatures called xenobots. So we call them xenobots. Xenopus laevis is the name of the frog they come from. What they do is they repurpose their little hairs, their little cilia that are normally used to redistribute mucus on the outside of the body. They use them to swim now. They've repurposed it. Here's it's chugging along. These guys, you can see the variety in behaviors. This one's going in circles. That one sort of patrols back and forth. Here are some collective behaviors. That one's going on a longer journey. These two are interacting somehow. These guys are just sitting there. All kinds of varieties. Now, I'm going to show you what happens when you put them in a maze. This is still water. There's no water movement here. There's no gradients. It's just spring water. It takes the corner without having to bump into the opposite wall. At this point, something happens, is some sort of nano free will kicks in or something, and it decides to turn around and go back where it came from. We have no idea why. There's some sort of internal dynamics that controls their behavior. So it's, you know, it's sort of exploring. It takes the corner at this point. Forget it. It's going back where it came from, right? So of course we need to now start to understand what drives these various behaviors. Now they can regenerate. So if you cut them in half, they can try to regain their new Xenobot shape. Notice the force that it has to generate through this hinge back here, about 180-degree hinge, right? Think of doing a bicep curl through when your arm is completely straight. It's really hard. And here they generate this massive force and they sort of fold themselves up back to a nice Xenobot. Now, here is the calcium signaling. Looks very brain-like. There are no neurons in here. This is just skin. Okay, these are just skin cells. And one of the most amazing things that they do is what we discovered is that when you take these creatures and you take away the ability for them to replicate in their normal froggy fashions, they can't do any of that anymore. What they do is they figure out a new way to replicate themselves. We call it kinematic replication, that as far as we know, no other creature on Earth does. They do exactly what we did when we created Xenobots. They work with the material they're given. So what we do is we sprinkle a bunch of skin cells, loose skin cells in their environment, and they've basically rediscovered von Neumann's dream. What they will do is they will run around, collect parts, and make copies of themselves out of those parts. Okay. Now, the reason this works is because they're not working in a passive medium. We are able to make Xenobots. I mean, the cells do all the heavy lifting. We don't know how to create a Xenobot. It works because the material itself has certain activities it likes to do, which means it likes to get together into a Xenobot and have certain behaviors, and they take advantage of that exact same property, as does evolution, in the material that they have. So they create these little clumps. The little clumps become Xenobots, and guess what they do? They go and they make the next generation of Xenobots, which makes the next generation of Xenobots. This is not a true evolutionary lineage. They don't have strong heredity yet, at least as far as we know. But it is a method of replication that they figured out within about 48 hours of life on Earth never having existed before. There has never been evolutionary selection for them to be able to do this. Okay, this has just never happened. This is completely novel. Where does it come from? And so we can start to think about this. There's a genome, a Xenopus genome. The Xenopus genome produces cells. These cells can work together to make an embryo, and it will look like this, and then make these little tadpoles running around. Or it can do this, and it will make a Xenobot. If we feed it and extend their lifespan for about 2.5 months, they have a developmental sequence. They make this. What is that? I've never seen that. I have no idea what that is. They have their own developmental sequence. Again, never existed on Earth before, as far as we know. And they have a completely different behavior, which is that. The Xenobots don't have a straightforward evolutionary backstory. The cells do, but they were selected to sit quietly on the outside of an embryo and keep out the bacteria. The selection pressure was completely different. The behavior is emergent. We don't know what their cognitive capacities are. We're investigating. So just to close up, I'm finishing up here. This is the framework that I'm working on. Called TAME, Technological Approach to Mind Everywhere. And the emphasis is this: there's a continuum of agency, no binary categories about whether something is cognitive or isn't, or has true memory or not. Everything is continuous. Empirically testable on this idea of what level of cognitive, it's basically Dennett's intentional stance, which is what applied to engineering, which is what level of cognitive model gives you the optimal prediction and control. Okay, not necessarily for brains. For any kind of substrates, selves are defined by the goals that they're capable of pursuing, and their intelligence is defined by the size of the goals they're able to navigate towards in abstract spaces. Developmental bioelectricity is a phylogenetic precursor of what we see in brains. It is the actual physiological medium for the software of life, and thus for the cognition of morphogenetic swarms. Other things may be too. I don't know. This is just the one that we found. And evolution is greatly potentiated by this kind of multi-scale competency architecture. And it doesn't find solutions to environments. It builds machines that solve novel problems, and understanding how that happens is one of the most exciting things for the future. So I will just close by reminding us that Darwin was impressed with the variety of living things, and he has this phrase, endless forms most beautiful. And I think many of us are going to be living in the world where all of the things that Darwin thought about, meaning all the natural evolved products of that one evolutionary tree on Earth, are all going to be a tiny corner of this incredibly huge option space. Every combination of evolved material, DNA, cells, tissues, some sort of engineered object, and nanomaterials, machines, and so on, and software, put together make a viable body with some kind of cognitive apparatus. We got hybrids and cyborgs and biorobotics, and just, you know, all of this is going to make the cantina scene at the beginning of Star Wars seem just absolutely tame by comparison. All of these things will be here. And the important thing is for society via ethics and other aspects is that we will no longer be able to know how to relate to something based on how it got here or where it fits on the evolutionary tree. It used to be easy. You could come along and sort of knock on something, and if you heard a metallic sound, you could say, that came from a factory, it's going to be boring, I have the right to take it apart if I want, and that's that. It's a machine. And if I do that and and it's sort of soft and fuzzy, then it's evolved and it's probably interesting and I have to be nice to it and so on. That's going to be out the window. None of those simple tests are going to work anymore. We're going to have to figure out a new way to relate to systems that we just have absolutely no guideposts to. So lots of papers about any of this. Please contact me if you would like them. I want to thank the postdocs and students who did all the work. Always thank the animals. They do all of the hard work and all of this stuff. Our funders, of course, the two disclosures, which are the companies that we're associated with. And yeah, that's it. Thank you so much. I'll take questions.
Thank you. Thank you, Michael, for your wonderful talk. I think we have learned a lot, from the lectures. Particularly, when I was going through your lectures, I find that it reminds me of a movie, the Amazing Spider-Man. So part of it is actually about the professor was like to grow sort of this regrow his hand on this movie itself. So it's very interesting. So I think that many of our colleagues have benefited from this. I would like to encourage our UC colleagues to discuss possible collaboration with Michael, and this will surely further strengthen our research collaboration relationship with Tufts University. So with that, let us move on with the Q&A sessions. And please raise your hand if you have any questions, and we'll do this on the first come first serve basis, or you can type your questions into the chat box. I will direct the questions to Michael for answering your queries. So let us now start the Q&A sessions.
We've got Brett Calcott has raised his hand if you'd like him to be able to talk.
Sure, go ahead. Brett, please.
Hi, can you hear me?
Yep.
Hi, yes, I can.
Thanks for a fascinating talk. I've been
Thank you.
looking at work for a while now, and it's very nice to hear it live. The question I have concerns the notion of a set point and a default set point. So you were looking at changing the set points interactively with the sort of living organisms, but as you mentioned, the default set point must somehow be encoded in a heritable way as well. So whereabouts, my question to you is, whereabouts, or can we find where those heritable default set points are, and then look at changing these heritable set points? So in other words, instead of intervening on the living animal, you could intervene on the single cell, the heritable single cell, and actually change a lineage at the genetic level.
Yeah, right. So let's see. So the first question is where do the default set points come from? So basically they emerge from symmetry breaking and amplification of an electrical circuit that's encoded by ion channel genes. So what evolution does, among other things, is sculpt the complement of ion channel genes that a collective of cells is going to have, that creates a circuit that by default, when the power comes on, so to speak, and the thing becomes alive, the electrical circuit settles down reliably to a default state. Now, if you wanted to, you could try to intervene there at the genetic level, and in fact, we've done that. We've added and removed channels, and you can certainly do that. The problem is, like with many of these systems, that there's a very indirect relationship. So you have to do a lot of computational modeling, and in fact, that's, for example, what we did in that brain study, to know what channel would I need to change genetically to give me the final pattern that I want. It's a really hard problem. We've made a little bit of progress, but it's very hard. I think in the end, a much easier way is going to be to take advantage of the memory and the behavioral properties of these systems and to actually train them. So I think, so yes, in theory, you can do it from the bottom up, but I think we're going to find that it's much easier to interact with them due to their computational properties, not at the level of hardware.
Brad, any more questions from you?
No, I think that's a really interesting. I mean, I guess the approach I'm coming from is I'm interested in the evolutionary pathways as well as the developmental pathways. So I'm like, I guess I'm really fascinated in how you can push these things in different evolutionary ways. But I think that the answer sounds definitely right. Trying to reverse engineer that kind of thing sounds like a very, very hard ask.
Well, and actually, I'll say one other thing about there's an interesting thing about evolution too, which is that if you think about evolution working on these, the fitness landscape, right? And evolution has paths from one region to another to try and create a particular kind of pattern. There's actually the evolution has the exact same options that we as engineers do. It can try to micromanage the process by tinkering with the individual proteins, right? The channels, and I'm sure it does that, but there's a much faster road. And I think actually what evolution does, given how incredibly rapid evolutionary change is, I mean, it's always shocking how fast this hill climbing search actually works in biology, is that what I think it actually tries to do most of the time is to exactly do what we do, which is to work at a higher level. So in other words, yes, you can try to reproduce a bunch of ion channels in just the right configuration to make specific things happen in the embryo, but it's much easier to tweak certain cells to stimulate those cells to kickstart an existing module the same way that we do. So it has the ability, right, to act at different levels, and both of those are available to evolution, and they have pros and cons, of course.
Thanks very much.
Thank you.
Thank you.
The next person is Paul Griffins. Paul, would you like to ask the questions? Or you would like me to read your questions?
You can read that. He's written it down in the chat. So
okay. So Paul is actually asking, your inductions of the eye can't help but bring to the mind or experiment on organizers, and the later discovery that simple chemical stimuli could induce specific structures and then later master control genes. So could you say something about how these previous discoveries of powerful interventions variables relate to your new discovery? How do these causes fit in a sequence of a network?
Yeah, thanks, Paul. Great question. Couple things. So it is certainly the case that bioelectric triggers are not the only triggers that exist. So there are so-called master regulator biochemical signals. There are biomechanical states that trigger specific things, right? So from that perspective, I think that evolution uses as what physics, it exploits physics in whatever way it can to create this kind of hierarchical modular control structure. Now, I will say that so bioelectricity is not alone in being able to serve as a high-level controller, but it has interesting and unique properties. So every once in a while, you'll see in nature or science, you'll see somebody made a logic gate out of diffusible molecules or liquid, some kind. And the reason it's in nature and science is because it's so hard. It is so hard to make these kind of computational devices out of biochemical states. Electricity is great for this. And I think evolution noticed how convenient it is as far back as bacteria, not because you can't do it another way. It's just super convenient to make feedback loops, to propagate information long distances, to make networks that do computations. It's just electrical network. There's no, it's not an accident that nervous systems use it, that our computer technology use it. It's just super convenient. And so also there will be two other kinds. So that I think is the more general, more interesting aspect. I will also say a couple of things. There is a so-called master eye regulator, PAC6, which you know, if you look in the developmental biology textbook, it will say this thing tells you where the eye should be. That only works in the anterior neurectoderm in frog. It does not work back, you know, behind the middle of the head. The bioelectric state works anywhere, and one of the things it does is turn on PAC6. Turning on PAC6 alone is not sufficient. The bioelectrics is further upstream. So I'm not claiming that in every possible case the bioelectrics is going to be upstream. There may well be cases where the most tractable, the most powerful control point may be biomechanical or bioelectrical. But in every case that we've looked at, it was the bioelectrics. And so I see your question about specificity. That's interesting. That's very interesting too. In the bioelectrics, where as far as we can tell, and we're open to surprises down the line, the specificity of the bioelectric code. In fact, we've done a lot of thinking about this, like where is what's the encoding actually? How do you say eye versus limb versus heart? Like where's the specificity? It seems to be, and we have computational models that you know, I'll show you offline that kind of implement this. It's in a spatial pattern of resting potentials. It is not in any one cell. There's no one cell that holds the pattern. It is, think of like they're almost like flags, right? Which is a defined, the flags of various countries are a defined piece of piece of rectangular real estate that has a certain pattern. And when you see that pattern, you know that that's mapped to a particular outcome. That's what bioelectric seems to be. Cells can recognize specific patterns. And we've been cataloging some of those patterns. We've been looking at how they're interpreted. That's how the bioelectrics encodes specificity.
Interesting. Any more questions from Paul?
We've got
We'll pass the floor to Alistair.
one more from Tom Close,
Oh.
if possible. Otherwise, we're good.
Sure. I can't see the questions. Do you have the questions, John?
Oh, I can see them on the Q&A. You want me to just
Yeah, if you
just do
click
it?
the Q&A down the bottom, it'll show up there.
Okay.
Yeah. So he's asking about the placebo effect. I think you're right on the money. This is something that we've spent some time thinking about recently, and there's some very interesting work on not only placebo effect but hypnodermatology and the idea of Albert Mason did some amazing work on hypnotic treatment of skin diseases, and we're not talking about stress-induced rashes, you know, that go away when you calm down. We're talking about actually missing cell types in the skin that were causing ichthyosis and various other things. I think you're absolutely right. I think as we start to think about levels of control, and okay, I can control at the molecular level and try to get all the gene expression correct to make something happen. That's brutally hard. I can try it at the level of at the tissue level and try to convince the tissue to do certain things. As one tries to climb that ladder, I think ultimately it's entirely possible that you can relate to these things from the nervous system down by convincing the master regulator between our ears that certain things are supposed to happen. It will percolate down to control the physiology of the body way better than we could micromanage it, right? I think that's entirely possible. So yeah, I think there is a version of the placebo effect here. And in fact, one of our projects is to take all of the concepts, or as many as possible concepts from cognitive neuroscience, and convert them into things you can do in the body. So there ought to be a version of the placebo effect that work. In fact, actually, I'll tell you, there is, now that you ask, it turns out we already have an example of this. I didn't even think of it that way before. We showed that in models of gene regulatory networks, you ought to be able to do, and when we're testing it now at the bench, but we don't have any data on it yet, you ought to be able to do associative learning. That is, you ought to be able to pair a neutral drug that doesn't do anything with a powerful drug that changes the state of the network. You present them paired, okay, the way basically Pavlov's dog, right? You present a neutral stimulus with a functional stimulus. You do that for a while. The network learns to associate them, and then eventually you can just present the inert drug, and the system acts the same way. that can be looked at as a cellular placebo effect, because at that point, you know, much as like we you know, people, human patients say, oh wow, he's giving me a blue pill, that's got to, you know, that's got to do something, you know, useful. The tissue does the same thing. Basically, eventually, once it's learned the pairing, the inactive drug causes the same effect. Maybe that's a version of a cellular placebo effect.
Thank you. Alistair?
Thanks. Yeah, I just wanted to, and that's an amazing idea, I think. Yeah, just want to check, Tom, if that's what you're after.
Are there any more questions, you know, from the floor?
One point you made fairly strongly, but briefly, was around control any external electric fields. You know, my colleagues and I do a lot of work with electrical stimulation and electrical impedance. We're looking at, you know, treatments for some conditions where we're, you know, applying external fields to the brain, to the spinal cord, to other things. Do you always steer clear of the external fields, or do you think they're another tool to use here?
There's certainly another tool. The reason we tend not to use them is for a very simple reason. Because we're not trying to control spiking in excitable cells, right? What we want to set up are standing, fairly complex patterns of resting voltage potential that stay for hours, in some cases days. We don't know how to induce that with electrodes, right? Because what you can do is now what's very good with electrodes is you can certainly control spiking, right? So you can trigger neurons, and you can induce directional electrical fields that will help cells migrate. So that's great. If you want cells crawling, so you set up your electrodes and they'll crawl. But the problem is, if I have a cell in you know, between two electrodes, and I set up an electrical field, this side of the cell will be depolarized, that side will be hyperpolarized, the net is unchanged. I don't know how to use electrodes to set up, okay, you're going to be -30, you're going to be -40, you're going to be -50, and you stay that way for 6 hours. I don't know how to do that with electrodes. It's entirely possible that someday someone, you know, if you have thoughts on that, definitely email me, you know, offline, and let's talk about it. I've never seen a way to do that with electrodes. That's the only way. There's nothing wrong with it. It's great for all kinds of applications, but I just don't know how to set up voltage gradients, like long-term voltage gradients with it.
Yeah, I think if you do, you end up changing pH and causing electrolysis, things like that. Mm-hmm. There have been some papers around using beamforming techniques, but if that really works, I'm not quite sure. You know, maybe in a bowl of saline, but yeah, something more complex. I haven't seen you know, it didn't move forward since a few years ago. So it'd be great to go back and take a look at that.
It would be great because a lot of the technologies, you know, let's say optogenetics requires you to have a light path to the cells, and that's great for things that are ectodermal on the outside, but what are you going to do, you know, deep inside? So yeah, it would be great to have those kind of technologies, you know.
There are some other things like the combination of ultrasound and kind of, you know, the lightning thunder kind of relationship.
The future, you know, this technology is, everything I've shown you is baby steps. You know, the future is going to be amazing. There's lots of reagents that need to be developed for this.
Fantastic. Thank you so much. It's been
thank you very much.
An amazing talk. I think it covered, you know, really kind of eye-opening, mind kind of expanding ideas. Fantastic.
Super. Thank you so much. I'm really glad.
We would like to wish Michael to continue to achieve greater heights in his academic career. And we hope that he can visit us soon, and we are happy to host him. And with that, I will close these sessions, and I wish everyone a good day.